So these are the various kind of agents which are there, and they are working, or they are responsible for various such activities. Then one more type of this is there, one more type of what we can say, this transduction mechanism which has been mentioned in syllabus. We will cover only what's given in syllabus. We will not go beyond it, because these they are very complicated, and we don't want to go for too much things. Okay. So one more pathway is given. Can you see this? Can you see this? Hello. Can you see this? So this is another kind. This is also one of the type of MAPKs. We have seen it in detail what are the MAPKs. So this is another version of class of MAPKs, mitogen activated protein kinases. And here the protein P38 is responsible into that, therefore it is called as a P38. This is responsible, or it is important in the aging and impairment or muscle, this one, or ultraviolet irradiations is responsible to stress stimuli. So many times it is also called as a stress activated protein kinase, SAPKs earlier it was called as a. These are again many varieties it has got, and it is having clinical significance, that is many places, many times oxidative stress is there. Whenever the metabolism is going wrong, or the normally free radicals are produced in the body, they are abundantly produced, but body has got a system to take care of it. There are some important free radicals, they are important in metabolism, but however sometimes they are too much activated and they causes a problem. And therefore because of this oxidative stress, this is called as oxidative stress, when we cannot have, they are very highly reactive molecule and they can react with any important this physiological structure in the body. So they are the most powerfully specific stress activating P38 MAPKs. This abnormal activity is there, that is higher or lower than physiological of, they are implicated in various stresses. It can be neuronal, bone, lung, cardiac, skeletal muscle, then RBCs, fetal tissues. So they are they are present everywhere, and if you know, you can see this SAPKs I told you, that is stress activating protein kinases or stress activating MAPKs are there. So earlier it was called as a SAPKs also. They are having a product of proto-oncogene, and RAS is a protein. So it can increase the activity of P38, and thereby it can causes high activity of transcription factor NF kappa B, that is nuclear factor kappa B, and because of that it can cause the changes in the genetic expressions. So some drugs are been found about it, or drug are been work, like pamapimode is a drug, on which clinical trials are there. Other drugs are having still code numbers, so they are still under studies. Then another is losmapimod is a P38 inhibitor, is investigated for the treatment of muscular dystrophy, which is one of the very debilitating condition. So this is one more problem it is having. Then we have already seen this C-Jun terminal kinases, that how this Janus kinases they are playing role, and how they are working through. They are important in the roles of DNA repair in the aging, and we have seen that how they are going for caspase and all activity. So this is how the Janus kinases or C-Jun kinases or JNKs, C-Jun terminal kinases, they are working. They are there. They are important in the DNA repair. We have seen that repair is very, very important. So these drugs are having various kind of enzymes. Particularly important enzymes are the enzymes which are causing phosphorylation of these various kind of molecules, like the sirtuin, stress responsive protein is there. So on that this amino acid serine is there on the 10th response, and this is helping to control the DNA damage. So this phosphorylation of this protein, this sirtuin on S6 facilitate the mobilization of this to DNA and it recruits the various enzymes like monophosphorylate polyribose polymerase 1, that is called as a PARP1. This enzyme is also very famous, PARP1, PARP2R. So these are known as a poly-ADP ribose polymerase 1. So these are also called as a NAD plus or ADP ribosyl transferases or poly-ADP ribose synthase 1. And this is enzyme which is important of the PARP family, 1, 2, 3, 4. This is a family. Okay, there are many in that, and their job is to clear the DNA breaks wherever they are. Okay. So this job is there, and they are responsible for control of this PARP enzyme. So this is how this C-Jun kinases they play important role in our functions. RAS already we have seen. Then lastly is protein kinase PIK3. We have seen them while MAPK also, that inside between these pathways this PIK is there, and this PIK, that is phosphoinositol triphosphate, is there, and they are important part in that. So these PIK3s are there. These kinases are important. They are present at various places. Again, this figure you will find, and you will find that here these PIKs are present. They are playing very, very important role in this signal transduction of various protein kinases, that is receptor tyrosine kinases, and because of that they are playing important role. And see, there are variety of genes are there which are responsible for this. They are very much important in many activities they are playing. They are having various activities like catalytic activity, regulatory activity, then other activities are there, and these all activities they are responsible for gene regulation by one or other men. So they are linked to various cellular function like cell growth, proliferation, differentiation, motility, survival, intracellular trafficking. There are many roles they are playing, and therefore they are activating protein kinase B. We have seen it as a AKT. If you remember, earlier we have seen protein kinase B is called as AKT in figure. The big figure which was there, in which all signal transduction pathways were given, in that it is shown as a this one. So AKT. So AKT and protein kinase B are same thing. Okay. So there are varieties of such pathway. mTOR we have seen recently in immunology. Okay, that is mammalian target for rapamycin. So this is they are all playing. We are now we are not still sure about what mTOR is doing apart from this immunological activities, but still it is doing important role. They are also key component in the insulin signaling pathway, and thus it is playing important role in diabetes mellitus. Will not go for in. They are also responsible. They are important in cancers. Then they are important in learning and memory, and there are many therapeutic agents nowadays are coming as a PIK inhibitors. There are many are there which are in pipeline, or they are coming. So this is how this third one, that is PIK3 kinases, they are playing important role. So here and lastly, now one more thing is there, that is various biosensors. So these biosensors are sensing various activities whatsoever are taking place. Okay. So these biosensors they will sense the changes whatsoever it may be. It may be antigen antibody interactions, it may be proteins enzymatic interactions and their outcome, then various receptors and their binding, then nucleic acid interactions, then the functions of organelles in cells, all the activities. So all these they will be sensed. There are several such biosensors are present in body, and these biosensors will be acting. They will be sensing what changes are there. We have seen them as a DNA repair mechanism. We have seen them as a function correcting measures. We have seen them as a feedback mechanism. So like that, these biosensors will sense various changes, and they will be providing the data. Okay, and these biosensors will thus help in activities. So these are the sensitive biological elements where they can judge organelles, cell receptors, enzyme, antibodies, etc. And it is a biological derived material or biomimetic compound. It interacts and binds, and then it will sense it. So there are several biosensors are available, and they will be sensing various kind of activities. So biosensor we can take in two ways. One is inside the body, which are sensing and giving the feedback signal is one thing, and second thing is artificially developed biosensors which will be sensing the various kind of activities, and the scientist can study what is going on. So there are biosensors, we can take it in two ways. Okay, so in both ways, one type is it is helping body to regulate the homeostatic mechanisms to the normal physiological processes or biochemical processes, is one kind of biosensors, and another one, as I told you, these are the instrumental ways, methods. Okay, so they will do it. In all cases, one important part there is that is known as a transducer. Simple thing is transducer means transduction, that is whatsoever information is there, that will be converted into another form which can be easily detectable in your other analytical system. Okay, and it convert into that. Suppose there is cell division, then this transducer will inform this what is happening. Okay, so this will transform the signal in the biological signal into physicochemical signals, and this physicochemical signal can be easily studied, easily checked using various kind of instruments. So there are variety of instruments are available physico-chemical way, and by that we can check it. So there we finish, there we complete the first topic, that is what we call it as a all cell related topic is over. I'll suggest you please go through this, read it thoroughly and make the questions. So all the dynamic cell, the structure and functions and components of the cell will be finishing. Please go through this well, because this is very new, very few people might teach this, and the depth particularly we have attained, I don't know whether earlier it was attained or not, but as per my knowledge goes, I have seen very less time that this much detail has been achieved. So I have tried my best to make it as simple as possible, and giving all information, nothing has to be left from the syllabus, but at the same time not giving you much details which are beyond your requirement. So at least the introductory information you will have about all these topics, that much I have tried my best. But these are, since these topics are that way very vast in nature, I'll suggest you that you go through it once, twice, thrice, and then accordingly have a discussion on it, have some question answer sessions. Okay. So tomorrow we may start new if time permits, or if not, then as usual next week we shall go for the classes. Okay. Or tomorrow I am planning to introduce you about pharmacological practicals, at least whatsoever there. These are two plans. Maybe we'll give some break and I'll give you some idea about various experimental things, because practicals are also assigned to me. So accordingly we may go for this. Let us see what we can do. Okay. So go through it. If you are ready with the next topic, if you are reading through the next topic which we have discussed earlier, you make the list of it, you make the list of reading about it, and then you put the further discussion what we want. Okay. So this is how we'll go for, and I hope you are understanding the things. I hope you will take the advantage of it and use it. Okay. So take care and be safe. જઈ हो जस्ट खत्म ज करे जस्ट क्लास खत्म ज कर्यो